Good morning, everyone. Like Dora said, my name's Crawford Baird. I hope everyone can hear me well, because I'm on my cell phone this morning. I'm having difficulties with my office phone, so if I'm getting soft or something, Dora, if you just holler at me.
Okay, that sounds good. I think you're really clear.
Okay. All right, so this presentation was originally designed for Georgia Pacific internally, just to bring awareness inside all of our mills to microbial influence corrosion. Because once we saw it and identified it in one mill, we realized it was in multiple mills. So we were trying to get the word out. And so there's going to be a lot of pictures and a fairly high overview of the problems we're seeing inside the mills. So just want to give you a brief idea of what the format's going to be. But for this audience, it's been slightly modified to that. On the first slide, I have one of my favorite quotes I ran across, and actually ran across this quote before I met Dora. It's by David White, who founded, and used to own Microbial Insights. And it's, microbial influence corrosion is an industrial venereal disease. It's expensive, everybody has it, and no one wants to talk about it. And that kind of sums up the experience I've had over the past 2 or 3 years with Nick. So, what we're going to go over today is just a brief process overview for everyone who's not in the pulp and paper industry. Some corrosion basics, just how these reactions work and what influences them. The corrosion we're seeing around Georgia Pacific and some of the different types of microbes we've seen. So, for those of you who don't know, pulp and paper, we pretty much take raw material, trees, softwood or hardwood, and convert it into paper of different sorts. Pretty much in all our mills, we're converting it into brown paper, some kind of bleached pulp, or tissue towel or napkin. And it's all the same process unless we're using recycle. But the process starts by us taking our raw material, wood, chipping it up, and taking the wood chips and actually putting it into a giant pressure cooker we call a digester. Heating it up to several hundred degrees and adding the chemicals sodium hydroxide and sodium sulfide. From there, we actually take the pulp out and wash it using water. And when I say that using water, is pulp and paper, the process is actually one of the most water-intensive industrial processes we have. So we use a lot of water in our loops, and we try and make those loops as closed loop as possible to conserve as much water as possible. So we actually wash all that chemical out of the pulp, and along with the sodium hydroxide and sodium sulfide, we actually wash out a lot of the organics in the wood that aren't needed to make the paper. So from there, you have two process streams. One is the pulp that actually goes out to, it can go to a bleach plant if we're making white paper, or just straight to a paper machine if we're making brown paper. Those bleach plants typically have some of the harshest environments that we've seen in our process, and a lot of that is we use chlorine dioxide, ClO2, to bleach the pulp. And some of those filtrates where we have ClO2 is where we've seen some of the highest microbial counts, which is kind of ironic, and it was quite astounding at first because we're finding corrosion, microbial influence corrosion in areas we thought that it would be impossible for microbes to live. It would actually, if you spilled some of that bleach filtrate onto the ground, we have chlorine dioxide monitors in a lot of our mills, the bleach mills. If you'd actually put that filtrate near a chlorine dioxide monitor, it would set the monitor off, and we thought it would be impossible for microbes to live in an environment like that, but we found otherwise. So that's one side, the pulp stream of the process. But on the opposite side of the process is when we're washing out the chemical and the organics. We actually send that to what's called our evaporator plant, where we evaporate the water out because the organic material inside what we call a black liquor has a lot of BTU value, so we actually burn it. So a lot of our mills they're pretty much a giant power plant with a paper machine attached to them. Georgia Pacific actually accounts for about 12% of all the biomass fuel, wood-based biomass fuel power generation in the United States. So, like I said, these are significant-sized power plants attached to a paper machine. So we evaporate the water from the black liquor, send it into a boiler to burn it, and we recover that sodium hydroxide and sodium sulfide chemical and send it down to our causticizing plant where we actually slake the sodium inside the liquor solution to get back to sodium hydroxide, because it actually comes out as sodium carbonate. I know that's a really quick process overview, but it's just to give you a general idea of the kind of chemicals we're dealing with and some of the environments we're dealing with. So we have a range of pH from down to a 2 up to about 11. or 12. So you can be quite different extremes of environments, and then temperatures ranging anywhere from ambient temperature up to a couple of hundred degrees. And through all that, we've seen microbes almost in every stage of the process. So it's been a real eye-opener to what kind of environments these guys can live in and the damage they can cause. So just a general introduction to corrosion. I'm not sure how familiar everyone is with it, but when you have a corrosion cell, there's two reactions that take place: the anodic reaction and the cathodic reaction. You can't have one without the other, and corrosion actually requires both of them. And the slowest reaction of the two will govern the speed of the total corrosion reaction. So if you're anodic limited, the anode will limit the corrosion, and vice versa. If you're cathodic limited, the cathode will limit the speed of the reaction. If you look on the left-hand side of the page, it just gives some typical anodic reactions. You have iron giving off electrons and giving off the iron ion into solution. And likewise, on the other side of the page, there will be a cathodic reaction to accept those electrons to complete the corrosion reaction. You can have it with nickel, and then the very bottom of the page is kind of just a general anodic reaction for any metal that is corroding. The anode is always the metal loss side of the corrosion reaction, while the cathode is always the electron acceptor. So just again, another look at the corrosion reaction. If you have iron bar and an electrolyte, the anode is giving off the iron ion. The electron is passing through the iron into the cathodic site. The electron can meet either a hydrogen or oxygen atom to complete one of these cathodic reactions, either oxygen reduction or hydrogen evolution, depending on the pH of the solution. That's just a real basic overview of a corrosion cell. And where this gets important is when we look at microbial influence corrosion, is that microbes are actually just changing this reaction. They change the kinetics of the reaction. They can change the speed of the reaction. They can change the corrosion product of the reaction. So that's really how the corrosion with microbes works, is they don't necessarily eat the material, eat the metal. They're just changing the existing corrosion reaction or corrosion mechanism. So we're going to talk briefly about corrosion influencing factors, because really, like I said, these microbes are just influencing a corrosion reaction. One of those is a concentration cell. So this is formed when a metal is exposed to an environment containing varying levels of electrolytes of either different substances or the same substance in different concentrations. That'll be enough to create a potential difference to drive that corrosion reaction. Or an oxygen concentration cell is formed when there is differing amounts of oxygen reaching different parts of metallic structure. Again, that can drive a potential difference and drive a corrosion reaction. And stress cells, formed when one area of metal is more mechanically stressed than another. The area of high stress is always the anode. And the reason I picked out these three influencing factors, because these are commonly seen with MIC, microbial influence corrosion. A lot of stress cells can be on welds of welded joints if they're not properly stress relieved. But more importantly, on oxygen concentration cells and concentration cells, is the bacteria can actually form these types of cells on the metal surface. Specifically, oxygen concentration cells, where you can get very, very low areas of very, very low concentrations of oxygen beneath the microbes on the pipe surface. That's particularly important when you're dealing with stainless steels, because stainless steels, the reason they're stainless is because they have a very, very thin passive film that is formed by chromium oxide on the surface. And if there is no oxygen present in the environment to replenish that chromium oxide, then it's easy for the steel beneath it to corrode. Actually, funny story, I heard this from a NACE instructor one time, that the reason we call stainless steels is back in the early days of the 1808 or 304, pretty much stainless steel. When they're first starting to deal with the alloy, when they were etching the alloy to look at the microstructure of it, they actually called it staining in Europe instead of etching. And they got to use the standard etchants for what we'd use for just a carbon steel, and it would never etch the surface. or it would never stain the surface to reveal the microstructure. So they started calling them stainless steels. So they weren't called stainless steels originally because they were corrosion resistant or wouldn't corrode. It was stainless steels because the standard etchants wouldn't work to reveal the microstructure. So I always hear a lot of people make the mistake of saying stainless steels won't corrode. Well, I'm here to tell you that stainless steels do corrode, in some cases worse than carbon steels. So what is MIC? MIC refers to the influence of microorganisms on the kinetics of corrosion processes of a metal, caused by microorganisms adhering to the surface, usually called a biofilm. And usually in that biofilm is not just one microorganism, it's a whole community of microorganisms. In a lot of cases, a lot of instances, they have a symbiotic relationship. They're using products of one species to feed another and vice versa. Or a lot of times you have anaerobic bacteria living beneath aerobic bacteria. The anaerobic ones are in between the aerobic and the metal surface in that low oxygen environment. So the term microbially influenced corrosion is used to designate corrosion due to the presence and activities of microorganisms. That is, those organisms that cannot be seen individually with the unaided human eye, including microalgae, bacteria, and fungi. Microorganisms can accelerate rates of partial reactions in corrosion processes or shift the mechanism for corrosion. So like I said, these organisms in and of themselves don't eat the material or eat the metal. They just accelerate rates of partial reactions. They can accelerate the anodic reaction or the cathodic reaction. One of those cathodic reactions we saw was hydrogen evolution. So some microbes, like methanogens, will actually take that hydrogen and pull it away from the surface to actually allow the rate of the cathodic reaction to increase, and that can drive the corrosion reaction faster. So microorganisms do not produce unique types of corrosion. Instead, they produce localized attack, including pitting, dealloying, enhanced erosion corrosion, enhanced galvanic corrosion, stress corrosion cracking, and hydrogen embrittlement. And I'm going to make another point here. The stress corrosion cracking, that tripped us up in the beginning. The first time I ever heard about MIC was in 2012. It was in one of our bleach plants where we had high chloride environments, and we were using 6 moly stainless steel, a super austenitic alloy AL6XN that was specifically chosen for the high chloride environments, but we started seeing leaks in the piping. And we would send samples out, and the metallurgist kept on coming back saying it's chloride attack and stress corrosion cracking, chloride attack, chloride pit. And we said, no, it can't be. It's not the right environment for us to be observing it. And it took us a while. It took us several months, almost a year, before we realized it was microbial influence corrosion. And it wasn't until after a year after we realized we had MIC that we actually found a test that positively identified the organisms present and tell us it was MIC. That's when we came across Microbial Insights. So what we need to have MIC, well, you've got to have an energy source, a carbon source, an electron acceptor, and water. And like I said, this is one of the most water-intensive industrial processes, so we've got plenty of water. Well, a carbon source and energy source, we've got both of those. We've got a lot of organics in our process just by default. We're taking organic material and converting it to paper. So we've got all the carbon and energy we need, and we've got the electron acceptor. So we're in the perfect environment for MIC, actually, in the pulp and paper process. So like I said, in the early days, we just kind of saw leaks on piping, and we didn't really know what we had. So I'm going to walk through several pictures of the symptoms that we saw, just kind of give you an idea of what we were looking at, and kind of as we're scratching our head, walking through this process and our thought process of identifying MIC. So we walked through a mill, and this is a 304 stainless steel water line right here, 150-degree hot water, and you'd see something on the bottom of a well that looked like this. You see what appears to be a leak in the bottom of the pipe. You start walking that line, and it looks like almost every welded connection on the bottom of the pipe shows some form or sign of corrosion. We think, well, surely when this pipeline was installed, we didn't have bad welds on every single joint of piping. So there must be something else going on there. And then on occasion, you would actually even see it in the stream of the piping, not on a welded joint. So we said, we don't know what that is. We don't think we had bad welds, but we're actually starting to suspect nick. Well, then in industry, we have to wait until an outage of some sort where this line to be isolated before we can pull a sample. So it wasn't until about 6 months later, after we first noticed the corrosion on the bottom side of the piping, we actually were able to pull a sample out of that hot water piping. And this is what we found. Actually, tubercles were going all along the bottom of the piping. And kind of closed up pictures of what they looked like. So we're still, at this point in time, we're still looking at these, not 100% sure if we had nick or not. And being fortunate enough to have several veterans in our company, or veterans of the company, I should say, working there, that have been there 20, 30 years. This site in particular had a history of poor water quality, a lot high manganese in the water. And actually, to have high manganese in the water, it doesn't take but about 20 parts per billion, 30 parts per billion, and they consider that pretty high for manganese. So we knew we had high manganese in the history, but we thought we remedied the problem. And so what we started to look for actually were manganese oxidizing bacteria, one of the first things we looked for. And with Microbial Insight's help and Dora's help, we actually found that we tested positive for the manganese oxidizing bacteria. We also tested positive for sulfate-reducing bacteria. And the sulfate-reducing bacteria, it's a common misconception with those guys that if you start doing any research on nick or hear anybody in industry talk about nick, everyone immediately jumps, it's got to be a SRB or SOB, sulfate-reducing bacteria or sulfur-oxidizing bacteria. Everybody immediately jumps to those two and says those are the ones, because what happens is they can actually, through their environment, create H2S and sulfuric acid. So everybody thinks, well, that's the most corrosive. If you don't see either one of those two species or that group of bacteria, you don't actually have nick and you're not getting corrosion. Well, we found that to be completely false. Sulfate-reducing bacteria doesn't necessarily mean we are or are not going to have corrosion. A lot of times what we see is just corrosion due to these manganese oxidizing bacteria, methanogenic or fermenters is what we found. And I've got more on that. I've actually got some metallographic reports showing what we found with manganese oxidizing bacteria that I'll show at the end. So this is kind of what we found after we took scrapings off some piping and sent it to Dora. This was another symptom we saw. This was actually something that the mill had known, been going on for a while. This is a hot water tank that feeds that hot water line. And about 8 years ago, I believe, the decision was made to build a carbon steel tank, and the plan was to just run it to failure because it had the same failure mode beforehand. So we've got a 250,000 gallon tank here that we knew we built, and we knew it would probably only last 10 years due to these tubercles, but no one really knew what they were. Like I said, we thought it was a manganese problem because that's what the history of the mill's water treatment was. But we've been treating that manganese with chlorine dioxide, oxidizing it, pulling out this solution. So the problem for the manganese wasn't there anymore, so it had to be something different. So immediately we thought it was nick here, or thought it had to be. And this was some of the frustration in the early days, too, that we ran across was the Nalcos, the Ashlands, actually Ashland Salinas now, so the Salinas of the world. We'd say, we plate something out for us and test it for nick. And they would plate, and they'd come back and say, you don't have any bacteria in your water. Your chlorine residual is good. And I said, well, I've got holes in my pipe and tubercles on my walls, so there's something in there. But they kept on saying over and over again, you don't have anything, you don't have anything. And what we found through this whole process, and Dora is the expert on this, is that actually something like 90+% of bacteria we can't actually plate and grow. We actually just have to do a DNA test to actually see if it's there, because you can't get the environment exactly right. But as you can see from some of these pictures, this tank was just completely covered in tubercles from the floor to the ceiling. Like again, this is just hot water. This is 150-degree hot water. And when we actually did the inspection of this tank, these tubercles over the past 5 years, or 6 years, have actually made quarter-inch pits on this half-inch thick plate wall. So we've gone through about half the tank life, a matter of about 6 years, due to the tubercles growing on the wall. So we sent those samples to Dora, actually sent 2 samples. And it's kind of ironic that the water right above the tubercles, but there's still water sitting in the bottom of the tank, tested differently from the tubercles. Didn't expect that one. But we found in the tubercles manganese oxidizing bacteria. In the water, we found traces of sulfate-reducing bacteria and methanogen. But in the actual colonies, it was mostly methanogen and manganese oxidizing bacteria. And then once we got Dora a little more involved in our systems, at this point in time, this is months and months and months after we first recognized we had MIC, we started looking into more and more species just as we learned about the system and looking at parts of our system to see what's present in each part. And the biggest one that was kind of shocked us was the fermenters, the big blue bar you see there. You can see how in the hot water tank we have extremely high concentration of fermenters as compared to the hot water system and almost non-existent cold water system. So although all 3 of those systems are fed by the same water, there's strikingly different concentrations of bacteria in each system. Again, this is just another chart showing the bacterial count in each system, as you can see. And obviously the cold water system would have the most because it's the most in that environment. And some of the different scrapings show different results, slightly different results. So scraping off the hot water piping walls versus the hot water tank floor showed some differences in the bacterial counts, just slightly different environments there. So then as we learned more about it, we started looking for it in other mills. This is a sample that came from another southeastern mill, and when we first got it, to the average person, you look at this and say, well, this is going to be under deposit corrosion. That's pretty typical. Just organic or inorganics and organics in the water caking up on the pipe wall, causing corrosion. And we said, well, does MIC play a role in this? And most people said no. We said, well, possibly. What about slime formers or anything else? Bacteria that can form on the wall actually create the surface conditions to allow inorganics to adhere to the wall. So we sent a sample to Dora, and we actually came back with several species of bacteria that were found. We never did any actual quantitative analysis on it, but qualitatively we did find that we did have bacterial species growing on the walls in that water system also. This piping right here in particular, it's actually one of the later ones in the slide deck. This is where we actually first noticed it. This is that 6 moly stainless steel we talked about earlier. It's a super austenitic stainless steel. It's very, very, very corrosion resistant. When we installed this piping and actually specified this grade for the chlorine dioxide or bleach filtrate environment, and within months of installing the piping, we noticed the piping was leaking. It shocked us, and it happened in multiple locations, particularly at the weld. And we kept on wondering what's going on. Well, we've done quality control on the welding, so we know it's not bad welding. Cut welds out and look at them, and they looked fine. So we're wondering what's going on. Why is our pipe leaking? And you actually get inside it, and you can see in here actual tubercle growth on the bottom side of the pipe. The rest of this is just hard water scaling. It's a calcium carbonate, what you see the yellow film here. Here's some tubercle growth along the weld seam. Right along the weld, you have one a little bit farther away from the weld. Again, another weld line right here. And then if we zoom in, this is the actual corrosion of the weld. So this is, like I said, 6 moly stainless steel. If you actually read Brenda Little, who works, I believe, at the Stennis Center and does naval research on microbial influence corrosion. She wrote a book called Microbial Influence Corrosion. She actually says that MIC on 6 moly stainless steel, just like this pipe in front of you, has never been reported. So this really made us scratch our head. We're reading, the experts with MIC, the PhDs and such that do this for a living, hadn't really observed what we're observing. So that really was another head scratcher. Then do we really not have MIC in the environment? Just some more pictures of the scrapings on the pipe. Scraping off your tubercles, you can see the corrosion beneath it. So we sent samples to Dora, and it came back, yes, we do have MIC. we didn't get the concentrations we expected, but we did have a presence of it, especially the fermenting bacteria. The sulfate-reducing bacteria was relatively low. And that's what everyone was telling us, is if we do have MIC, it had to be a sulfate-reducing or sulfur-oxidizing bacteria. We found those concentrations relatively low. Again, this is bleach filtrate here. So it has a high chlorine dioxide concentration in it. And that, like I said, that chlorine dioxide concentration is high enough to set off a CO2 monitor. And chlorine dioxide in a lot of applications can be used as an oxidizing biocide. So it really shocked us that we would be getting MIC in these environments. After we didn't find much with on the previous slide showed of high concentrations, we went back and we looked at other forms of bacteria in our system. And one of the higher ones we found were the denitrifying bacteria. You see some of the highest counts were in the raw water and the white water. Excuse me. Again, the sulfate-reducing bacteria remained relatively low until you get over here to some of the filtrate lines. We saw a little bit higher concentration there. And fermenting bacteria were one of the highest here in the standpipe and some of the filtrate. So, what we've learned and seen is that the environment doesn't really matter. These microbes can adapt to different environments, grow in a lot of different ones, whether it be hot water or bleach filtrate. And I wish I had pictures in here, but we actually had some black liquor, which is a chemical we use in the pulp and paper industry. It's a byproduct of cooking I talked about earlier. It's that sodium hydroxide, sodium sulfide, sodium thiosulfate, sodium sulfide in a lot of organic material. And we actually see MIC in that, and that can be anywhere from 140 to 180 degrees. We've actually seen it inside the tank systems there. So it doesn't really matter the environment. We still seem to find it, detect it, and it just causes us headaches. This was one of the latest mills. This was actually, it's not technically a Kraft pulp and paper mill. This is a recycle mill. So the big difference is they don't have the wood inlet or the digester and the cooking chemicals of the sodium hydroxide and sodium sulfide. This one's taking recycled paper and bleaching it and turning it into another consumer product. So this is what we began to see on our piping systems on the inside. Actually, what we see at first was just like the other mills. You see localized corrosion on the external of the piping system. And you wonder, what's going on there? And then you wait till an outage and actually go inside the piping system when it's time for the inspection. And what you saw on the weld line here was found. We thought, we've got MIC here too. And Dora actually made a trip up to this mill to look at some of the systems to help us identify the species of bacteria that were there and kind of help us come up with process remediation, what biocides to use, what can we do to treat this, because it was a very extensive problem. The tank you see in front of you is actually, I think this tank was 6 to 8 months old and was already starting to show signs of MIC. What was astounding is that a lot of these mills, some of these were built, I think one of our oldest mills was built in 1904. The newest mill was built in about 1984. But up until several years ago, or a few years ago, we didn't really know this was a problem or never noticed it as a problem. And especially didn't have its tanks and heat exchangers and piping systems start to form leaks within 8 months of service. So the real question is what's changed and why are we all of a sudden seeing this problem? Now, one thing that has changed is a lot of the alloys we use. We seem to find it's a lot worse with stainless steels versus carbon steels. As we've upgraded our metallurgy, our problems have upgraded too. So spending more money doesn't seem to have helped us very much. Again, this is the same symptoms we see on, a lot like our piping systems. We see the same things on our tanks. It doesn't really matter what the mill is or what the process is. It's always the same symptom, something on the weld line of a tank or a piping system. Starts to show signs of very, very small leaks. And then once we get on the inside of the piping system, you can see where tubercles have grown all along the weld line. And this one actually is on these tanks here are actually on a paper machine involving white water and stock. And and things. So it's 140 degrees. It's got plenty of pulp and organics in there for the bacteria to feed off of. But still, it's somewhat of a harsh environment when you think about the temperature of the chemicals, the bleaches that may have been added, and biocides that are added to paper machine. You wouldn't expect to find bacteria growing on your vessel walls. So here's the bacteria reports we actually got from Microbial Insights. And these were actually some of the highest concentrations we found across the company to date. As far as the sulfate-reducing bacteria was present in high concentrations of methanogens and fermenting bacteria. And like I said, this was another vessel that was only there for about 6 to 8 months. We started to see signs of corrosion on a vessel that you'd expect to last 20 years with no problems. So that's a really quick overview of some of the problems we've seen inside the company. And I know I've skipped over a lot of information very quickly, just in the interest of time. I know we've only got about an hour plus questions and things. So sorry if I went through that very quickly. But there's also, I wanted to show some metallurgical reports. Because like I said, in the early days, we had early stages of identifying the problem. We had a lot of problems just trying to identify what we actually had. So this is a picture of a piping spool piece that came out of one of the mills in the hot water system. And we were trying to identify if we had nick or not. So what we actually saw was a spool piece that actually had a leak on it on the weld, just like we saw in all the other cases. And so we actually had a chance to remove this one without an outage, so we could plan and just do it anytime we wanted to. So that's why we pulled this sample out of the line. It seems small and irrelevant, but it's actually one of the keys to us finding the solution to our problem. So we pulled the sample out of the line. This is all we could see from the outside, but we assumed we might have nick there. So we sent it for metallurgical analysis, and they pulled it apart, cut it up. And this is actually what they found, one little tubercle growing on the inside. And when we did metallurgical analysis on it, this is what the microstructure looked like. So this is what kept on throwing everyone for a loop, is we would do a metallurgical analysis, and what we find is something that looked a lot like chloride stress corrosion cracking. So everyone would come back and tell us, you've got chloride problems, you've got stress corrosion cracking. This isn't nick. This is just some kind of underdeposit corrosion or something like that. But what you find, actually, if you read more and more in depth into the literature, especially in this case where we've got manganese oxidizing bacteria, is that the bacteria actually create a very low pH environment and attract chlorides to it. Form heavy metal chlorides in that environment that even though the base water, the water outside the microbial colony, is extremely low in chlorides, like 13 parts per million, which is very, very low, especially for our industry. You can get extremely high concentrations of chlorides beneath the tubercle and actually cause the corrosion you see in the picture in front of you. So this is a 304 stainless steel spool piece, and generally you could say that thing's rated for a couple hundred parts per million of chlorides in 100 to 200 degree or 100 to 150 degree environment. And so we're in a 13 ppm chloride environment with 150 degree hot water. We'd say, hey, we should be fine. We shouldn't expect to see any corrosion, but we do. So that just goes to show how that microbe changed the conditions beneath its colony in such a manner that it would corrode the metal surface. That was one of the stumbling blocks, I guess, for us in the early days as we're learning about microbial influence corrosion and how it worked. Is that although it looked a lot like chloride stress corrosion cracking, the root cause would actually be the microbes growing on the surface. I've got one more analysis to show. This one is this is the AL6XN, the 6-moly super austenitic stainless steel. So this would be where we see a leak in a pipe and wonder what's going on. Send it out for metallurgical analysis, and they could actually see the nodule. But what they found was actually a poor weld, so a lack of root penetration welding. So we had these guys come back and tell us, hey, your problem isn't nick and your problem isn't chlorides. Your problem is you had poor welding. Well, we say in most cases we actually X-rayed and did a lot of quality control, so we know it's not poor welding. So it's got to be something else. Now, poor welding didn't help the situation, but it surely wasn't the root cause of the corrosion we're seeing in the piping. For those of you not really familiar with the weld, this would be a butt weld here, one piece here and one piece here. And you actually lay a root actually in here, and then you steel the weld out to the surface of the piping system. So this will be the inside diameter of the pipe, and this will be the outside diameter of the pipe. And what you see here is where the metals actually did not fuse together. They form a little crevice to form crevice corrosion. So that's what this company actually came back and would say, oh, you've got crevice corrosion, just because they found poor welding. Even though poor welding was true, this crevice corrosion over here didn't cause the pitting over here. So we actually still had microbes growing on the inside of the piping surface. Like I said, I know that was a very, very, very quick overview of some of the problems we've seen and some of the microbes we've run into in a very basic overview of pulp and paper process and corrosion. But with that, I was just going to open the floor for questions, because really we're still we're learning about microbial influence corrosion, just like I'm sure those of you on the call are learning about it. So we are by no means experts in it, but are still learning and welcome any help or advice from anyone out there in the field.
Well, thank you very much, Crawford. That was an excellent presentation, very, very interesting, and I learned a lot of what you've been doing in the process, even though I've been involved in this. So I appreciate you doing this for us today. So if you can enter questions that you have for Crawford into the GoToWebinar software, we'll be glad to answer those for you now. So the first question, Crawford, we see pretty dramatic differences between the water and the scraping samples. So do you have recommendations of whether based on what you've seen in these mills, that you'd recommend to look at the water, the solids, or do you recommend both?
I definitely recommend both. Because, even if you see it in the water but you don't see anything on your piping walls or your tank walls, then it may not be hurting you just because of the metallurgy or process flow conditions. And likewise, if you see it on your walls, it may not be necessarily in your water. And what that would tell you, say if you're seeing a lot of tubicles in your water on your surfaces but nothing in the water, what may be happening is when you're actually taking your process down for an outage or some kind of maintenance window, especially if it's extended for several days, the water actually becomes the breeding ground at that point as it gets old. As it's set there and it's cooled off to ambient conditions, so that's when these microbes may be taking root, is actually when you're down for a shutdown and not necessarily up in operation. And once they form their little community in their hut in there, they can actually withstand some of the environments of your operation. So it may help tell you if you've got microbes forming from actual normal process conditions, or if it's from some kind of a shutdown you've done that's been extended for several days.
Okay, great. What changes have you implemented from the information that you've gained from this?
There have been several. At the mill where I showed the manganese oxidizers, a little more history on that mill, we noticed it on some heat exchangers. And so the mill was built in 1984, and we had carbon steel, plain carbon steel heat exchangers at the mill. And as when they thinned, actually, they didn't have pitting corrosion, they just thinned over time. And so in the early 2000s, we actually went back and replaced one of them with 304 stainless steel. And then I think it was about 2008 or 2010, we replaced another one with a higher grade stainless steel, a 2205 duplex stainless steel. And last year, we'd actually seen signs of corrosion on the 304 stainless and the 2205. So one of the changes was not necessarily to always upgrade the metallurgy. We actually changed out the 304 stainless steel heat exchanger and went back to just a plain carbon steel, which is actually a cheaper alternative and seemed to last longer. So we kind of learned that maybe the higher metallurgies, more expensive metallurgies, aren't always the solution. Some of the other things we're starting to be aware of or trying to make everyone aware of is when you design a process, be sure and design a high enough flow through the piping system to try and help resist the colony's growth on the piping system. So if we get a high enough velocity of water or whatever we're pumping through the piping system, the colony may not have an equilibrium site or a good site to actually grow, so it may prevent it just by doing that. in I guess in one of the other environments, the one with the CO2, Dora, you've been involved in some of these conversations with us where we've been talking about biocide, what biocide we want to try and use now. And like we've seen, the oxidizing biocides haven't worked, so we need to go to some other type of biociding to help protect our system, but we still haven't found out what we want to use for that one yet.
Okay. What about sample collection? Do you find it's easy to collect the samples for this analysis?
Yes. As long as the process isn't running, it's very easy. What I've done historically is either just open up a sample port and pull water out, or what other substance I'm testing, and depending on what it is, I maybe freeze it to stop all microbial growth and then ship it to Microbial Insights, or either scrapings are very easy. Just put them in a sealed container and ship them out. So it's very easy to collect samples and give them to you all. And I don't know if you'll have any other specifications you like around your samples other than something like that.
Yeah, no, I think you covered it really well. Sorry.
As far as getting a sample to test, I've just tried not to contaminate it with the outside environments. Just scrape it off and seal it if it's an actual hard sample, and if it's a liquid, and especially water-based liquid, freeze it and then ship it out.
Yeah, I think the key thing is making sure you keep it at that cold temperature to kind of minimize those microbial changes in transit after you collect the sample while you're transiting it to the lab. And then just, like Crawford was saying with sample collection, make sure that you're using gloves for collection so you're not contaminating it with anything else. All right. The next question is, have you been able to reduce or control MIC through any changes in the raw water treatment?
We have not. And like I said, we've had discussions with the Nalcos and the Salinises of our industry, because usually we have our processes are large enough, for those who don't know, that we have our own water treatment plants. We usually keep some kind of chemical rep on site 24/7 as far as to monitor how we're treating our water. And what we've been getting from them is that your chlorine residuals look good, your chlorine residuals look good, and they don't really have any other recommendations for water treatment other than that to control organic growth. So we haven't made much progress on changing how we're treating our water to prevent it, but it's still a work in progress, and we're just trying to work with the chemical vendors to figure out something that hopefully will fix our problem and probably make them money too. So.
All right. A couple of people have asked Crawford if it's possible to get a PDF of your slides so they could review them along with the audio presentation on the website.
Yes, I'll convert it to a PDF and send it to you, Dora.
Okay, that'll be great. So if anyone wants that, just email me and I'll send that out to you. The next question is, what, if any, correlation does microbial activity have with carbon steel and the hydrogen sulfide to form elemental sulfur? Would we have any MIC that you think would be associated with this reaction?
None that I know of. I've seen, it appears in our process that carbon steel is the most resistant to it. And I said we got a lot of sulfur in our process, but we didn't really just find an extreme amount of the SRBs and SOBs. So I really don't have a lot of experience talking with that. So I really don't know. But what I do think is going with it on the carbon steel, stainless steel, MIC basis is that with a stainless steel, what we get, and I didn't really discuss this one very much, is called the size effect. And the size effect comes into play with the size of your anode versus the size of your cathode. And so if you have a very, very small anode and a very, very large cathode, and again, you've got the electron transfer, that's a current, you've got a very, very high current density on the anode, so you'll drive deeper pits quicker. Whereas vice versa, if you had a very, very small cathode and a very, very large anode, the current density on the anode in that case is extremely small, so you don't necessarily get the very deep and quick pitting. So the size effect plays a big role into it, and I think that comes into play with the stainless steels, because you're going to have a steel that's completely passivated in almost all the areas, but underneath the microbe you actually have almost zero or very little passivation. So you get an anode right beneath the microbe, and the rest of the steel becomes a cathode.
Okay. Well, thank you very much, Crawford, for your time today. I really appreciate you spending time, doing this webinar for us. I know you're really busy, so we appreciate that you're taking time out of your schedule for that. And it was an excellent presentation. So I'll send out the PDFs to people as soon as we have those available, and we'll get it on our website so that you can review this. Then if you have more questions, feel free to email me, and we'll get together and get with Crawford and get those questions answered for you and send them out to the group. But again, Crawford, thank you very much. You did an excellent presentation. You're a great speaker. It was a pleasure to have you here today.
Thank you.
All right, thank you. Thanks, everyone. Goodbye.